Jenny Arzalwata, second year college, taking up Bachelor of Secondary Education, and I'm going to discuss about the mutation and gene regulation. The mutation and gene regulation. DNA, genes, nucleotides, alleles, and mutation. DNA is the chemical name for this long, stringy stuff that sits inside of cells. Genes are segments of DNA. Most genes code for proteins. Nucleotides are the building blocks that make up DNA. Alleles are a different version of the same gene. There are small differences in their nucleotide sequences. Mutations are DNA copying errors. When they happen in egg and sperm, they are the source of new alleles that can pass of offspring. Mutations are changes in the genetic sequence, and they are main cause of diversity among organisms. Mutation can be inherited. Parents that have mutation can pass them to their offspring. Germline mutations, present in every cell of an individual, including the egg or sperm used in the production of offspring. De novo or new mutations occur by chance in one of a few eggs or sperm, or just after fertilization, and are not present in every cell of a parent. This explains situations where a child has a genetic disorder that is unseen in the family history. Mutations acquired environmental agents, called mutagens, can alter DNA. An example of a common mutagen are the UV wavelengths in sunlight associated with skin cancer. Acquired mutagens are typically not passed to offsprings but can be in the altered DNA sequence in egg or sperm. So we have the before the incoming UV light and the after. So the first types of mutations is the point mutation, a change in a single nucleotide. So we have the original sequence and the point mutation. Deletion changes the number of DNA bases by removing a piece of DNA. Insertion. One or more bases are inserted into the original DNA base sequence. This is sometimes called a frame-shift mutation because it shifts the codon reading frame. And also we have the type of mutation, the duplication, consists of a piece of DNA that is abnormally copied one or more times. This type of mutations may alter the function of the resulting proteins. Inversion is a type of mutation where a sequence of nucleotides in DNA is reversed, the original and the mutated chromosome. Gene regulation includes a wide range of mechanisms that are used by cells to increase or decrease the production of a specific gene product, proteins, or RNA. Gene regulation can occur at various steps. The amount of products depends on rate of messenger RNA synthesis. Transcription is the first step in gene regulation. It involves copying a gene's DNA sequence to make an RNA molecule. mRNA degradation. Each mRNA refers in the rate at which they are translated into a protein and in the stability of mRNA molecule. The longer lasting an mRNA molecule is, the more protein products that can be transcribed from the mRNA sequence. So protein synthesis, translation, the process by which a protein is synthesized from the information contained in a molecule of messenger RNA, then a transfer RNA molecule carrying the amino acid methionine binds to what is called the start codon of mRNA sequence. So we have the example of gene regulation. So enzyme induction is a process in which a molecule, drug, induces, initiates, or enhances the expression of an enzyme. The lac operand is an interesting example of how gene expression can be regulated. Viruses despite having only a few genes, possess mechanism to regulate their gene expression, typically into an early and late phase, using collinear system regulated by anti-terminator or splicing modulators or the HIV. So that's all for the mutation and gene regulation.